I now call to order the Society's 2420th meeting and the annual President's Lecture in the 149th year since the Society was founded on March 13, 1871. Good evening, everyone. My name is Larry Milstein. I'm the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members and guests to tonight's lecture by Joachim Frank in the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C., including our members and friends around the world who are watching the live stream of tonight's meeting on PSW Science's YouTube channel. We will begin with a few announcements, followed by a reading of the minutes of the 2419th meeting and the lecture by Jack Gilbert on the World Microbiome Project and Integrated Microbiomics. We will then turn to this evening's lecture, followed by a question and answer period. When the question and answer session is done, I will present a thank you gift to our speaker, make a few closing announcements, and adjourn. Thank those who make PSW possible, and adjourn the meeting to the social hour. First, please join me in thanking the sponsors of the 2019-2020 lecture series, the Policy Studies Organization, in cooperation with the American Public University, and a generous donor who was asked to remain anonymous. Please also join me in thanking the sponsor of tonight's lecture, the intellectual property law firm, Millen, White, Solano, and Brannigan, where I'm a partner. Please also thank our sponsors of another type, the volunteers who have dedicated their time, expertise, and care to carrying on the work of the Society without compensation, serving science and the public, and maintaining the almost 150-year-old traditions of the Society. Please especially join me tonight in thanking one of the members of the General Committee and the crew who keep this organization running and growing. A special thank you to Robin Taylor. PSW's corresponding secretary, who, in addition to attending to email and other correspondence, including the lecture announcements and dinner invitations, updates and maintains the PSW membership database, takes care of many bookkeeping tasks, updates the website content, sets up the video equipment and packs it back up, directs the live stream, does the post-production, and posts the edited videos to YouTube and Vimeo. And she does a lot of other things as well, and keeps the rest of us focused and in line. She is truly the XO of the organization, responsible for continuously improving its operations. Thank you, Robin, for your many contributions to PSW. I think we can leave her up for a couple of minutes here. I am pleased to announce that the following new members have been elected to the Society. Anthony D'Amico, a statistical programmer and independent consultant interested in demography, who learned of PSW from Eventbrite. Andrea Borns, who is here, a microbiologist with ATCC Federal Solutions, BEI resources interested in microbiomics, tropical and vector-borne disease, including schistosomiasis, Zika, and dengue fever, and in phage therapy, who comes to PSW through PSW member and phageologist, Carl Merrill. Thank you, Carl. Marcia Lucas, a psychologist in private practice interested in neuroscience, human behavior and cognition, and psychoneuroimmunology, who comes to PSW through the live stream of the 2,419th meeting and the lecture by Jack Gilbert, and Marcel Horowitz, a physician interested in general medical sciences and urology in particular, who comes to PSW through the Cosmos Club, and tonight's speaker, Joachim Frank, whose background and interests will be clear to you in part from tonight's proceedings. Please join me in welcoming them to membership. There is a signed reprint of Volume 1 of the PSW Bulletin for all new members. If you haven't received yours, please see me after the lecture during the social hour to pick up your copy. And if you purchased a PSW ribbon, please see Ann McQueen at the ribbon table. Well, she's over here now, but yeah, please see that woman over there to pick yours up. She'll be at the table in the back. Recording Secretary James Heelan will now read the minutes of the 2,419th meeting and the lecture by Jack Gilbert on the World Microbiome Project and Integrated Biomics, delivered to the Society on February 7th, 2020, right here in the Powell Auditorium. James.
Thank you, Larry. Hi, good evening, everyone. On February 7th, 2020, in the John Wesley Powell Auditorium of the Cosmos Club here in Washington, D.C., President Larry Milstein called the 2,419th meeting of the Society to order at 8:04 p.m. He announced the order of business, that the evening's lecture would be live streamed on the internet, and welcomed new members to the Society. The minutes of the previous meeting were then read and approved. President Milstein then introduced the speaker for the evening, Jack Gilbert, professor in the Department of Pediatrics and the Scripps Institute of Oceanography at the University of California at San Diego. His lecture was titled, The World Microbiome Project and Integrated Microbiomics, Understanding Microbial Populations and How They Shape the World. Microbes mediate the health of humans and other organisms. The human immune system developed to control our exposure to microbes, keeping away the harmful and fostering a welcoming environment for the helpful. A disruption of either the immune system or the microbiome will also disrupt the other. By better understanding this bidirectional relationship, scientists can produce actionable interventions for targeted impacts. Gilbert studies the relationship through microbiome-wide association studies, identifying the association between microbial activity and human health and disease. For example, microbes living in the colon ferment fiber and produce acetate, a chemical that facilitates the brain's synthesis of leptin, which controls satiety. Hyperphagia can therefore be treated by ensuring a healthy gut and consuming elevated levels of fiber. Because microbes can produce amino acids, Gilbert's team, in cooperation with Cedars-Sinai Medical Center and the University of Accra, have experimented with supplementing children's diets with cellulose. Compared to a control, children in Gilbert's sample had fewer incidents of stunted growth and mental delay. Gilbert's team has also identified relationships between the presence of certain gut microbes and liver cirrhosis and chronic diarrhea that Gilbert said can be resolved through fecal transplants. Gilbert then described an experiment his team conducted with the genetically related Amish and Hutterite communities of the American Midwest. The two groups had the same allelic genetic sensitivity to the development of early onset wheeze and childhood asthma. But the Amish have only 3 to 4% asthma, and the Hutterites have 25 to 30% asthma in the respective populations. Gilbert's team examined the immune systems of members of each group and determined the Amish have healthier immune systems. Gilbert believes the Amish benefit from living near their farming activities, which expose them to a diversity of microbes at young ages, while the Hutterites place themselves at a disadvantage by living further away from their farm activities and limiting the members of their community who engage in the activity. Gilbert described experiments exposing germ-free mice to the fecal microbiomes of Amish and Hutterite community members that he said confirmed his hypothesis. Gilbert has also done experiments exposing germ-free mice to the microbiome of normal mice that challenge traditional notions of instinctive or taught behaviors and behaviors associated with depression. Gilbert is now researching the effects of the microbiomes of people with various levels of social interaction. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 10:08 p.m., President Milstein adjourned the meeting to the social hour. Temperature, 3 degrees C. Weather, partly cloudy. Attendance here in the Powell Auditorium, 102. And viewing through the live stream on the PSW Science YouTube channel, 27. Respectfully submitted, James Heelan, Recording Secretary.
Thank you, James. Do we have any comments or corrections to the minutes? If not, I will entertain a motion to accept the minutes as read. I have several motions. Do I have a second? All members in favor? All members opposed? The minutes are accepted unanimously as read and will be posted to the website in due course. We now turn to tonight's lecture on single particle cryo-electron microscopy, revolutionary methods for determining structures of biological molecules in their native states. And it is my pleasure to introduce tonight's speaker, Joachim Frank. Joachim is professor of biochemistry and molecular biophysics and of biological sciences at Columbia University. Previously, he was professor of biomedical sciences in the School of Public Health at the State University of New York at Albany, and senior research fellow at the Wadsworth Center. Joachim's research has been on developing techniques of electron microscopy and single particle reconstruction of biological macromolecules, specializing in mathematical and computational approaches. His research notably led to the development of single particle cryo-electron microscopy, greatly facilitating atomic resolution determination of molecular structures in their native states, leading to a revolution in the structural study of biological macromolecules and of their dynamics. He was awarded the 2017 Nobel Prize in Chemistry for this work, together with Richard Henderson and Jacques Dubochet. In addition to the Nobel Prize, Joachim has received many other honors and awards, including the Franklin Medal for Life Science and the Wiley Prize in Biomedical Sciences. He is a member of the National Academy of Sciences and of the American Academy of Microbiology, a fellow of the American Academy of Arts and Sciences, and the American Association for the Advancement of Science. He earned his PhD from the Technical University in Munich. Please hold questions for the question and answer period at the end of the lecture, and join me now in welcoming Joachim to the podium.
It's a great pleasure to be here. Thank you very much, Larry, for inviting me here. And I confess that I didn't know about the existence of the society, and I had never heard of the Cosmos Club. So these are great discoveries that I make tonight, and it is a great pleasure to meet you all. So I'm going to tell you about the technique that I helped foster. And to begin with, I would start with the job of what we really want to know. We want to know how a cell is organized, but we actually want to know how life functions are constituted in the cell. And these are constituted by the interactions of molecules. We want to know how they interact. We need to know their structure. And obviously, the cell is an unbelievably complex system, and we cannot really study molecular interactions in the cell per se and hope to get to high resolution. So what we need to do is we have to accept a step of reductionism. We talk about a system in isolation. We have to extract it from the cell and then hope to be able to infer what is actually happening in the cell. So this is essentially a giant step ahead here because we are looking at simulations that have been done much more recently about systems that we have gained a lot of knowledge of. But the main point of this slide is really to introduce you to Bruce Alberts, who came out with an article in the Cell, 1998, where he popularized the concept of molecular machine. So essentially what he said, we have always underestimated the cell. The cell was thought to be a place where molecules essentially sort of interacted at random, like billiard balls. And but instead, what he wanted to emphasize is that there is actually a form of organization in which many molecules interact with one another in a processive way. Okay, processive means there's a before and after. The ligands interact with some kind of a molecule in a very choreographed fashion. And so what we really ideally want to understand how these kinds of systems work. And I told you about reductionism. We study a subsystem in isolation in vitro, hoping to approximate the processes in the environment of the cell. Now, in a number of cases, including the ribosome, it's been possible to actually run an entire system, like the molecular machine can really run in vitro and perform all the functions that it performs in the cell. And such a system is the ribosome. And the ribosome, for another reason, is one of my favorite systems that I spend a lot of time with. Okay, so these are examples for molecular machines where we have a lot of knowledge, meanwhile, and they are all processive. They involve a lot of binding of ligands. They are highly complex. And an example where we have many states of a molecular machine is the ribosome. This is just the tip of the iceberg. There are many more steps in these processes, and in order to understand the entire system, we need to know all these steps and the intermediates. So what I'm going to tell you about is, first of all, what I call the ancient history, EM and X-ray crystallography, and then the single particle techniques. The concept emerged in 1975, and then the first single particle reconstruction, which was still using negative stain. There was no cryo initially. And then the first, the Cryo-EM reconstructions 1990 to 2012 with increasing resolutions up to 5 angstroms, all already made with ice embedding. And then from 2012 to now, we are experiencing what is called the resolution revolution, where we have, where we now are able to get with the single particle Cryo-EM to resolutions that parallel the resolutions obtained with X-ray crystallography. And so today we have an exponential increase in cryo-EM structure depositions, so much so that we in a few years, we sort of match the depositions that have been taking place over decades from X-ray crystallography. And then a few new things that I've been focusing on in my lab. One of them is time-resolved cryo-EM, and the other one is mapping of a continuum of states using cryo-EM. Okay, so maybe some people have heard of hybrid meetings. Hybrid meetings have only been convened more recently that brought together X-ray communities and cryo-EM communities because there's a lot of interest sort of in cross-fertilization between the two camps. But the point of this slide is that there were hybrid meetings already at the time when I was a graduate student. Here is in Hirschek, 1968, there's a workshop on X-ray crystallography and EM of proteins organized by my mentor Walter Hoppe and Max Perutz. Max Perutz was one of the pioneers of protein X-ray crystallography. And then a few people you might recognize by name that were at these workshops were Harold Erickson, Richard Henderson, who got the Nobel Prize with me, Ken Holmes, Hugh Huxley, and Nigel Unwin. So these kinds of meetings were typically with something like 50 people, 50 to 80 or so, and the main purpose of it was really skiing, and interrupted by workshops in the morning and late at night. And here are a few shots of these meetings. So I have to essentially, I'm realizing that this is a very very general audience, so I'm going to tell you about X-ray crystallography and then Cryo-EM in order to get the main introduction. So in a crystal, you have many copies of the molecule which are arranged in regular order. And with the exposure to the X-ray beam, we get a diffraction pattern. And so this diffraction pattern encodes information on the structure, but it doesn't encode everything of it. It doesn't encode the phase. So the phase determination is sort of a separate problem, but this problem has been solved. So the X-ray beam must be high intensity. The crystal must be almost perfect. And to date, to bring everything in context that I'm saying here from now on, 140,000 structures have been solved by X-ray crystallography, and they're all available in the public database. And they form the basis of essentially what we call molecular medicine. So the knowledge is really mostly created by X-ray crystallography. So what's wrong with X-ray crystallography? Well, one of them is, which is sort of a basic criticism, is that the crystal packing means that the molecules are not visualized in all conformation and binding states that are important for function. And actually, the structures that are in the database, they might not be very meaningful in terms of understanding life functions because they have one particular conformation which is determined by the overall minimization of the energy of the crystal packing in the so but more important is that many molecules do not form highly ordered crystals at all. And what about those? We don't have any structures of those. Now these happen to be very, very important molecules receptors, ion channels, and so forth. And so there is a huge, huge gap in the database that I told you about. Sample quantity can be a problem because when you do a crystallization experiment, you need multiple quantities of a sample in order to try out all kinds of conditions for crystallization. And so you might very soon run out of sample. Okay, electron microscopy. It can be used to solve molecular structures as well. And here, actual images are formed, not just diffraction patterns, images. And they are at very high magnification, for instance, 30,000. And to reconstruct an object, many different views must be collected. But there are negatives to this. The sample must be very thin. Electrons are readily absorbed by matter. And electrons also strongly damage the molecules. There is a need for low dose. And 10 to 20 electrons per square angstrom is typical of what needs to be used. And the consequence of this is that the images are very noisy, the so-called shot noise, because of the random incidence of electrons, which simply means that you cannot take the images at face value. They're simply too noisy. There is a need for averaging. Okay, so here we're talking about the beginning of molecular electron microscopy. And the real heroes here are David de Roussier and Aaron Klug at the MRC, and they were the first to use the computer in order to do three-dimensional reconstruction of a biological object. And this object was a bacteria phage tail. Now, bacteria phage tail happens to have helical symmetry, so it is essentially in the application of a certain method of retrieval of information, which is unique to helical structures. So they worked out the theory on how to do this for helical structures, but it was essentially a math and computational programs that were tailored to a particular geometry, okay? And also a very ordered arrangement. Here's another another structure was looked at by Tony Crowther at the MRC, and these are icosahedral structures. Many viruses, such as the coronavirus, have icosahedral symmetry, where 60 units are arranged in very regular order. And he developed a mathematical system to retrieve information from such a structure. Again, you have a mathematical system and computational programs that are tailored to a particular geometry. Here's another one. In this case, we have a flat crystal, a two-dimensional crystal. It's very thin, a crystal of bacteriorhodopsin. And there, Richard Henderson and Nigel Unwin were the first ones to develop a way of retrieving information from this. And they came up with the first structure of this 7-helix bundle in this membrane protein. Okay, you see what all of them have in common. All of them have in common that they use the fact that molecules are in regular order. And then they also have in common that the programs that were developed were unique. They were tailored to particular geometries. So it was not, there was nothing general in this approach. So my mentor, Walter Hoppe, who was actually an X-ray crystallographer who developed an interest in electron microscopy. So he posed the question, why crystals? Why can't we just, look at molecules as they are? And so he developed what is called electron tomography. And what you do is you have the molecules situated on the grid, and and then you tilt the entire grid in regular intervals, and then take this entire data set and use methods of 3D reconstruction to reconstruct the entire sample, the entirety of the grid and the molecules on top. Okay, so you get several reconstructions of several molecules all at the same time. So the big problem is that the accumulated dose exceeds 1,000 electrons per angstrom square when you use this kind of method. And what it means is that you do get a reconstruction, but the reconstruction is totally meaningless because the molecule by that time has burned to cinder. It has suffered so much radiation damage that nothing of the original structure is left. So in this environment in which I sort of grew up as a scientist, I thought this doesn't make any sense. Okay, so I developed my ideas in contradistinction to my mentors. So I give him a lot of credit for that. So why crystals? I also posed the question. So I came up with a single particle technique. The structural information comes from images of single, which means unattached molecules that exist in many copies. Well, the reality is, if you take a cell extract and purify it for a particular molecule, then you get thousands, you get millions, you get billions of molecules, and they are all the same, okay? And they occur in random orientations in the sample. So there is one advantage, which is that they're also free to assume all naturally occurring conformations, which is different from what we know from the crystal. And the molecules are randomly oriented. What it means is that we don't have to tilt them. They already tilt themselves. So which means that a single snapshot may already give us hundreds of particle views. And as we collect more and more snapshots, more orientation will be covered until we have enough for reconstructing the molecule in 3 dimensions. So the whole concept is very simple, okay? And from my PhD thesis, I already knew that we can use cross-correlation function in order to align images with very high precision. I found this out by looking at electron micrographs, images from the electron microscope that were taken of the same specimen, but one after the other. So there's a lot of noise built up in between. And I was able to show that those very noisy images can be aligned with very high precision within something like 3 angstroms. So that gave me hope that maybe the same can be done with not the entire micrograph, but pieces of the micrograph that contain a molecule view. Okay, so this was in my dissertation at the Technical University in Munich, and they published this now 50 years after I, so 49 years after completion, and it's called Analysis of High-Resolution Electron Micrographs Using Image Difference and Reconstruction Method. I was very flattered that they believed that 50 years afterwards, it still had some kind of value. Okay. but they are now, I have an entire stack in my office. So the idea that I just formulated about single particle reconstruction, I put on paper in 1975 in ultramicroscopy, and I made specific reference to what I showed you before, to Unwin and Henderson, who had reconstructed the bacteriorhodopsin and the 7-helix bundle. So the tenor of that paper was really, couldn't we do the same thing with single molecules? You know, do we need crystals? And so at that time, I was at the Cavendish lab, and I was working to some extent with Owen Sexton, who is also, he had a focus on image processing. And together we developed a formula. The question was simply, you know, do we have the right kind, do we have the sensitivity to even localize molecules under the assumption that we have them embedded in ice, and we make assumptions about the size of the molecules and the contrast that we achieve and so forth. Does it even work? Okay, so we convinced ourselves. We developed this formula here, which we call detection equation, and this formula essentially told us that yes, it's going to be possible to get to atomic resolution if we are able to get molecules embedded in ice, and if the molecules have a certain size, you know, beyond a certain size limit. So this was very important in terms of motivating us to go forward. Now, Owen Sexton decided not to follow in this whole line of research. So far so good. I had spelled out in this ultramicroscopy paper, you know, what could be done, but I had no way of telling, you know, how exactly to do it, because there were a whole number of problems that needed to be solved. Align images, okay, I already knew it's going to be working, but then how to estimate the resolution of the average, how to sort and classify images, how to find projection angles. Well, this was really the biggest problem, finding projection angles, and then reconstruct in 3D from completely random orientations. So essentially, I had a concept, which was a great concept, but I didn't have any tools to put it to work. And so I had to essentially create a whole workbench to deal with these problems. And that's what this stands for. I had to create a workbench for myself so that I was able to essentially play with all kinds of ideas without each time recreating a new program. Okay, so I needed something like a modular image processing system, which was made up of individual units. Okay, so these was a modular image processing system, which was called SPIDER, and they had hundreds of operations, and these were all operations which I would call primitives. They are essentially problems that could be solved as a unit, like Fourier transform was such a unit, rotate, shift, all these things are sort of trivial, but you have to put the workflow together, put them all, assemble them, and then you can assemble them in many different ways. So many people can participate in these kinds of game, and once such a program system exists. So now I have to introduce the ribosome. Very important. at a certain time when I gave talks about these ideas, and I didn't have really pictures, images to use, I ran into Miroslav Bublik from the Roche Institute in Nutley, New Jersey, and he approached me and he showed me these pictures here of ribosomes from HeLa, from human ribosomes, from human cells, and he asked me, would they be useful? Could you do your, could you try your programs on these? And I said, sure, they look marvelous. And so that's how I got into the ribosomes, okay? I was a physicist. I knew nothing about biology. I knew nothing about the relevance of ribosome in life functions. And so I sort of learned this along the way. And the great thing about the ribosomes was that nothing was known about them. Very, very little was known. It was known how it made proteins, given genetic information, but how it was doing this structurally was completely unknown. And so Jim Lake was one of the people who divined the structure of the ribosome just by staring at micrographs. So there was nothing computational about it. He just stared at them and he said, and he thought, well, this kind of model would probably would probably give us these kinds of projections if I tilted them around. Okay, so so he came up with this model here, which actually existed in three-dimensional form. He distributed them. It has a little copyright notice on them, and I have one in my lab. So this was the knowledge about the ribosome. So given the pictures that I got from Milas, I set out to apply the kinds of techniques that I had been developing. And here, this was just not three-dimensional reconstruction, but just two-dimensional averaging. But I showed that I could get reproducible results from doing this with different subsets, and the results were really fantastic. They were appreciated. I got a publication into Science, and I got my first grant proposals. So, but there's this big problem, which is the problem of heterogeneity. We don't want to have to look at an image and then make decisions of, this is a good molecule to use in these kinds of tasks, and reject others. We want the computer to do this, and so that was a big problem. How do we sort images by using computational techniques with quantitative criteria? And it so happened that we found an answer in 1981, and this was by using multivariate analysis of aligned molecule images. Now, multivariate statistical analysis had existed already in clinical laboratory science for a very long time, but it was applied to one-dimensional vectors which coded for blood samples of patients. And we just so happened that we saw these programs producing clusters. And then we thought, well, gee, if it can work with blood samples, why not? Why couldn't it work with images? And so we were able to essentially import our aligned images into a system that was designed for analyzing blood samples. And we got this beautiful clustering out of this. So there was a real milestone in the development of this entire technology. And then comes the very big problem is how to find the angles of projections via some kind of bootstrap method. And we came up with the idea of the random conical tilt reconstruction. I'm going to tell you in a minute about this. In 1979, I had the idea, and it took 7 years to put it in practice. And so the idea was here on an overhead, and to make it a little bit easier, later on I was helped by a scientific journal to help me to do a real professional presentation. So the idea is really that if we have molecules that are aligned in a particular way on the grid, they're all in the same orientation on the grid, then the only degree of freedom that they have is that they have different azimuths, okay? But that doesn't give us any new information on the grid. But if you tilt this kind of arrangement by a high angle, by 50 or 60 degrees, then these projections that we get are genuinely different. They are three-dimensional, and we get information from them. It is as if in the coordinates of the molecule, it is as if the electron microscope were walking around in a cone. Okay, and we call it random conical because these are not just equally spaced, but they're random because these azimuths are random. Okay, so this was the idea, and it took some time to put it in practice. And the hero here is Michael Rademacher, one of my postdocs, who got an education at the same place as I, with Walter Hoppe. So he was really specifically trained in three-dimensional reconstruction algorithms, and so he made it possible to get to a realization of this idea. So then the first reconstruction was of a 50S, which means the large ribosomal subunit from E. coli ribosome. And here it's displayed at different threshold levels. And it came out in 1987. Actually, the first publication was in 1986. So this was the first single particle reconstruction that was ever done. And I'm very proud of it. And then later on, initially we had a representation of this as a stack of contours, which was one of the ways of representing three-dimensional information. So this is mounted in a stack, as a stack of contours mounted in a wooden box. And when later on the Nobel Museum in Stockholm approached me to bring something idiosyncratic that had to do with sort of the invention or discovery, so I thought this was the best gift that I could give them. So it's displayed now in the Nobel Museum. So right here, I'm presenting it, and next to me is Jacques Dubochet with a very primitive precursor of a freeze plunging instrument. And here is Richard Henderson presenting an atomic model of the bacterial rhodopsin. So that brings me to this real big revolution which happened in 1981, but it took a number of years to really permeate the scientific community, which is the invention of the cryo method, okay? So everything that I had developed before had nothing to do with cryo. It was done for negative staining, which is a very inferior method of staining molecules. But now came a very big revolution, the invention of the frozen hydrated specimen preparation. And Jacques Dubochet got the Nobel Prize with me. So, in a nutshell, we put the specimen on a grid. We pipette it on a grid, and then an important step is a blotting. We need to blot the excess of the liquid away in order to get the right kind of thickness, and then the grid is plunged into liquid ethane or liquid propane at liquid nitrogen temperature. So this whole technique already existed and was developed by Bob Glaser. And the very big difference is simply this introduction of liquid ethane. Because without the liquid ethane, if you plunge the sample directly into liquid nitrogen, then air bubbles form between the sample and the liquid cryogen. And these air bubbles prevent fast heat transfer. And since the heat transfer is not fast enough, it allows crystals to form. So the water actually becomes crystalline ice, the kind of ice that we know. And this ice expands, it's crystallized, it damages the molecule, and it's a very poor way of preparing molecules. So the introduction of the liquid ethane meant that the sample could be plunged and have virtually instantaneous heat transfer. And under these conditions, the water forms a different kind of ice, which is called vitreous ice. It's glass-like. It has almost the structure as it has in liquid form, and the molecule is not damaged. So this became a very big revolution because it meant that molecules could be now embedded in a form that was close to the form that they were used to. Okay, so this was a workhorse that we had since the year 2000, the so-called Polara, which we had in Albany at the time, and then later on, because of the success of the entire technology, it was replaced by a very, very big, high-powered instrument, which is all embedded in a box. The plunge freezer, here are different models of the plunge freezer. You can build one manually for something like $200, or you can buy one for $60,000. So it's your choice. And well, you know, there's a difference because in one case you don't have any control over the humidity or the temperature and so forth. So the results, it can be different from one day to the other. So that's a big difference. So here is an image of ribosomes recorded on film. And I have to just insert one picture, which is, I told you how to get the 3D structure of molecules with this random conical tilt reconstruction. But this is something like a bootstrap reconstruction. It needs to be refined. And so there's a refinement kind of loop that we have to go through. So, but then we were all very happy because now we had, for the first time, a method that could produce images of molecules that had never been seen before because they could not be crystallized, and they were artifact-free because they were done with the with the vitreous ice method. And so this was the first time that we got really incredible images for the ribosome, for all kinds of hemocyanin. This is just one of the species of, of hemocyanin, and the calcium release channel. Everything at very moderate resolution, but still it was sort of a breakthrough. Now at that time, nobody thought that this method would ever get to atomic resolution. There was just too much stacked against it. But I'm just going to show you a number of things here. In 1995, the E. coli ribosome, which enabled us to put together a model on how protein synthesis would work, putting the mRNA and the tRNAs in place, and suggesting the way the polypeptide emerges from the large subunit. And in fact, we were very successful in mapping, in subsequent experiments, in mapping tRNAs, elongation factors, and so forth on the ribosome at the various stages. And we came up for the first time with a structural model for the elongation cycle. An elongation cycle is a cycle that the ribosome has to go through for adding an additional amino acid to the nascent chain. So if we have a protein that is 200 amino acid long, the cycle has to go for 200 times, okay? But this immediately and then made its way into textbooks. So then came a real big breakthrough when it turned out that by using very powerful programs of maximum likelihood, it was possible to actually extract several structures at the same time from the same sample. So we could really have a mixture of molecules, or we have a molecule that had different states, different conformational states, as it is needed in the work cycle, and we could all extract them separately from the same experiment. It was unbelievable that could be done. And I called it a story in a sample, you know, because I got very excited about it. It meant that we could get all this reconstruction out and then, you know, put arrows in between using some additional information from biochemistry and so forth. But there was really a story in the sample. So this is an example where we see different states of the ribosome all intermixed, and now we were able to pull it out. And so this essentially indicates that it was a very long road now in this entire development, and it's by no means we were the only contributors. There were many groups have then worked on various developments of the methodology. But there's a particular reason why I stop here at 2013, because that's a particular time in which we were really stuck at a resolution of 5.5 angstroms. We couldn't get any further. We added images, we classified and so forth. We never got beyond this point. And it was all because the recording was on film. And just about that time, in 2012, the first commercial developments came on the market of very powerful electron detectors. These were detectors that could detect single electrons. And they had an incredible large signal-to-noise ratio. And with this, all of a sudden, the resolution revolution occurred. It was a complete game changer. Now we are no longer confined to 5.5 angstrom. We can get to 2 angstroms. Somebody even got to 1.3 angstroms recently. Okay, so it's really unbelievable. So these are the different modulation transfer functions for the different media, and you can see comparison between the film and these powerful cameras. At the Nyquist limit, you still get a sizable signal. So now, compare with what you saw before. I showed you a recording on film, and so here you can see so much more contrast on the ribosome. So we and many other labs have now started using these cameras. And one of the first projects had to do with looking at an elongation factor G mutant bound to the ribosome. And you can see the quality of the images that we obtained. I can't go into the details of this here. But the best resolution that we have so far obtained for the ribosome is 2.5 angstrom. And it even has places where the resolution goes all the way to 2.3 angstroms. And you can see the quality of this, of the different chemical groups and the base pairing. So I'm just showing you, give you an impression of this three-dimensional structure and the complexity of it and the incredible amount of detail that comes out of this. So when I started with this whole development, I never thought we would get into this kind of era. Okay, we're sort of going through here and to show the different resolutions. And the very high resolution is in the interior where you have a lot of stability. Here's another example, the AMPA receptor, which is a receptor in the brain. It's now possible to get these structures of ion channels by surrounding these molecules by some kind of lipid package that simulates the membrane environment. So the molecule feels like it is in the membrane, so one can really get the structure. This was one of the most difficult things to do with X-ray crystallography, and now it can be done in a wide range of structures. I mentioned the ryanidin receptor before, or the calcium release channel, and now previously we were at this kind of resolution, 30 angstrom resolution. Now we got to 3.6 angstroms, and we have it in different stages of its functional cycle. Cystic fibrosis. Here's a group at Rockefeller that for the first time got an atomic structure of the cystic fibrosis related ion channel. And we are also working on this. We are working on the mutant, which is implicated in the disease. In this slide, I just chose this slide in order to show you the what we see at 2.2 angstrom resolution. We actually see the rings, and at 1.3 angstroms, we actually see already the beginning of hydrogen atoms. So it's really unbelievable. And I told you about the number of depositions that are happening now. these are the number of released structures per year. So it's really the rate of depositions. X-ray is pretty much now sort of settled. NMR slightly going down, and Cryo-EM is going up, going steeply up. So it's a it's a factor of 10 every 5 years. So impact in biology is really particularly in the domain of ion channels, receptors, but also very large molecules such as spliceosomes, which have an intrinsic flexibility and are very large and exist in many stoichiometric varieties. So here, at this point, I'm thanking collaborators. There are many collaborators that have been instrumental in making this whole work possible over the time, and they're in two different groups. One is in the ribosome specialty, and the other ones are people in methods development, mathematicians and computer scientists and so forth. And then my wife is really has had to put up for 37 years with a German professor, okay? So I have a few slides, if that's okay, to talk about new technologies, as I promised before. One is time-resolved cryo-EM. The objective here is that we look at short-lived states that only live between 10 to 1,000 milliseconds, and those cannot be seen with the kind of very slow specimen preparation that I have introduced before. And so it involves a microfluidic chip. And the first devices were developed at RPI and Wadsworth Center using silicon. And now we're using organic polymer, PDMS, together with the Columbia Engineering Department. And these are the people that have been working with me at the engineering department and also my own group. And so the idea here is that this is a chip, and we have two solutions that we want to add together. You mix them very fast in a mixer, and from that on, they go through a reaction channel. And the reaction channel has different lengths depending on the chip, and they are designed for different times, okay? So you can have a chip for 200 milliseconds, one for 500 milliseconds, or whatever. And in the end, the product is sprayed onto the grid as the grid falls down. So this is the apparatus as it's implemented right now. And I show you a biological process that's just been published that's been done with this, and it shows that it really works as specified, and it can be used really for any molecular machine that you can imagine. So here, this is the process. It's very, very simple. After the whole protein is made, a stop codon is encountered at the A site of the ribosome. Now the stop codon cannot be read by a tRNA. There is no tRNA to read it, but it has been recognized by a so-called release factor, either RF1 or RF2. So they recognize the stop codon and then lodge on the stop codon on the ribosome, and then they're supposed to reach the peptide and chop it off right here. Okay, and then the peptide is released, and then we go on in the whole process. So this is now a process that was not very well understood, and the big puzzle was that the RF1 or RF2 the X-ray structures show the RF1, oops, sorry, that this factor is very compact. It is so compact that when it binds to the stop codon, it can never reach the peptide where it's supposed to chop it off. Okay, so that's a big puzzle that suggested that perhaps it binds first to the ribosome, and then once it's bound, it sort of stretches it out and makes a big conformational change and reaches the place where it's supposed to act. So this is what we are supposed to see. We want to see the compact form and the extended form. Okay. So here's the reaction. The reactant number 1 is the termination complex. Right here. Reactant 2 is RF1 or RF2. We mix them together, and then what we see after 24 milliseconds is indeed the compact form sitting on the ribosome. So this is a short-lived state. It's never been seen before, 24 milliseconds. And when we wait for another 40 milliseconds, it's extended. So between 20 milliseconds, 60 milliseconds, we see this reaction taking place. Okay, it's beautiful. It works. It works exactly as designed. Okay, here they are next to each other. And the other technology, I have to go through this very quickly, is mapping continuum of states. Well, it's sort of, the concept is essentially simple. If you have a lot of molecular images, like hundreds of thousands, millions of them, then that ensemble covers so many states that even covers states that are only visited by a few molecules. So essentially you get a continuum. You get a continuum of states. And if you have a continuum of states, it's essentially a multidimensional continuum of states. You can sort of put it, represent it in a low-dimensional space, and then you have reaction coordinates that you can associate it with it. And then you can map the energy landscape, because energy landscape only you can infer from the number of observations in each state. Okay, so this is the first time that we are able to map the energy landscape of a molecule. Now, if we are able to do that, then we can do everything. We can understand how a molecule works. Okay. So essentially, this conformational continuum is in a high-dimensional space, and what we need to do is design mathematical methods that stretch out this manifold into a low-dimensional space. And then in this low-dimensional space, we can actually then observe the various conformations. So these are the people that I've been working with, and very important are the people at University of Wisconsin, at Milwaukee, Abbas Urmast. And the first application was with a ribosome. We had 850,000 Cryo-EM images. And what we observed was that when we did the mapping, we observed was we saw a complete closed pathway in the energy landscape, and we found that by going along here, we found ribosome conformations that we already knew from experiments before that were associated with the elongation cycle. So it looked like, with this set of images, which were not in no particular state, they were naked ribosomes. They had no tRNAs, nothing, no mRNA, no tRNAs, and so forth. They're naked. So it turns out that when they are in this thermal environment, they essentially visit all the conformational states that they would have to visit if they were actually producing something, okay? So I'm thinking about it as ribosome dreaming, okay, dreaming. What would I do if I had only tRNA and mRNA to play with, okay? So that was very interesting. It really showed us that there's a conformational freedom of the molecule itself, which is sort of a product of evolution. It's not just a structure, it's really this conformational versatility that has developed over time, which is designed in such a way that it picks up energy from the thermal environment in a productive way. And then calcium release channel is another one in which we have applied the method. And this is a little bit more too complicated to explain, so I just wanted to conclude now. So far from settled and complete single particle Cryo-EM is a dynamic field which will have many surprises in the future. And time-resolved Cryo-EM can give detailed kinetic information about the reaction of a two-component system on a time scale of tens or hundreds of milliseconds. And the ability to retrieve multiple conformations is a particular forte of single particle reconstruction, as it promises to reveal the functional dynamics of a molecule. Okay, so I conclude with this. We are here assembled on the roof of the Black Building at the Medical Center. I need it. I don't? You do? I do, okay.
So, thank you very much. We have time for some questions and presumably some answers. There is a procedure that we have, which many of you know, but I'll repeat it here for those who don't. There are 3 microphone runners, I believe. There's a red one, a blue one. Do we have another one? And a black one. And they will come to you in the order, red, blue, black. Or maybe we'll do black, red, blue. Okay, and when you get the microphone in the order, I'll tell you what the color is that's up, and you stand up, tell us your name, tell us if you're a member, and ask a question, save dialogues and polemics for the social hour. So we'll start with the black microphone here. That would be Carl Merrill. Carl.
Well, first, I really want to thank you. When I was an undergraduate student, my professor, William Guy, had studied with Rutherford when they worked out the structure of the nucleus of the atom. But he was convinced that the inside of a cell was a colloidal substance that we would never penetrate. And I was convinced someday we would do something like this, although I had no idea how. I'm only sorry he couldn't be here to see this lecture. But the question I have is, what I see in these pictures is that you can see the macromolecules, but a lot of the energy, of course, comes from ATP and that sort of thing. Will we ever get to the level where we can actually see that too, see how they interact to impart the energy to this?
Well, not in a not really in a continuous motion, but we do see with that kind of resolution, we see the interaction of GTP and ATP. So we see different stages of this, but we cannot claim to make a movie out of this that have any reality to it.
The red microphone. Oh, blue microphone.
This is kind of a general question.
That's Tim Thomas, who is a member.
Oh, Timothy Thomas. I am a member of the society here, but it's all right. Sir, as I understand, and I could be totally wrong, the ribosome is present in every cell and necessary in order to translate from the genetic instructions to protein. And that because it's present in every cell, they have used it to do the phylogenetic tree of all life. See how it varies as it has evolved in the course of life. So how can it be so variable that you can, how can it be so variable that you can tell the tree of life from it, yet it has to be essentially the same darn thing in every cell? And you've only done these in just a few species. How much does it vary?
Yeah, it varies a lot, but it varies at points that are essentially unimportant for the basic functions. The basic functions are really what I would call a core, the core of the ribosome, which have really been essentially unchanged. And then what I would call bells and whistles. There are all kinds of things that might have to do with specialization of ribosomes. There are many sort of tentacles that are going out. There are RNA expansion segments. Very little is known about what the function of these are. And then you have specialization of certain surfaces of the ribosome because they are specialized for the interaction with the membrane. And so they are more important for eukaryotes than for prokaryotes.
I could maybe add a clarification to that a lot of the phylogenetic mapping of microbes on the basis of ribosomes is actually on the basis of 16S ribosomal RNA. And it's only on the basis of certain variable regions in the 16S RNA. Not quite done yet. So, red microphone.
Adam Jacobson, I am a member. So in your historical overview, you mentioned that X-ray crystallography is not suitable for certain classes of molecules because you can't get a crystal out of them. Are there similar kinds of limitations on cryo-electron microscopy?
Yeah, that's a very good question because the big problems are still in sample preparation, in getting samples prepared in a essentially sort of a routine way. Sometimes you have proteins that like to associate with the air-water interface in a particular direction, so then you don't get all the orientations. And sometimes they aggregate, they form aggregates, and then it's a big question on how you get the single molecules. So there are many problems that can occur. So I would still think it's far from routine. But if you have something like a ribosome, then you know what you're dealing with, and then you can essentially do it in a very routine manner. Yeah.
The magenta microphone.
Yes, I'm Joe Foreman, and I am a member, a life member, as a matter of fact. I saved a lot of money by living a long time. But my question is, you mentioned the damage due to the high-energy electrons on the sample, but now you seem to have overcome that. I did not understand how you avoided that damage.
Yes. I mentioned when I said this, I mentioned that it can only be overcome by averaging. So you notice that I'm talking about tens of thousands or hundreds of thousands of ribosome images. These are many more than would be required from a geometric consideration. So since you have such an abundance of signal, you will get beyond a certain signal-to-noise ratio. And so this is the only reason, because of the requirement to use a very low dose.
Any other questions? Blue microphone.
Yes. My name is Ryan. I am not a member, but I work in the field of medicinal chemistry, and it's important to have structures of crystals with the endogenous ligand bound, and oftentimes ligands don't have a high enough affinity or have a high residence time in the active site or binding pocket. Does the time-resolved cryo-EM pose a solution for getting weak affinity ligands, getting, images of those bound in?
Well, I'm not sure about this because you still have the problem of the low stoichiometry, but what at least in the standard kind of cryo-EM, what you can do is you can do classification. We can essentially put a mask on the place where the ligand is expected, and then sort images on the basis of whether or not we see material there. So we can really extract a subset of molecules that have the ligand bound.
A red microphone.
Will Angel, I am a member. Thank you, Dr. Frank. Could you talk a little bit more about the costs of cryo-electron microscopy and how much they've come down and how much you think they'll continue to come down? Thank you.
I can't talk about the latter part, but the former part, it is very expensive. The microscope itself is like $5.5 million, and then the camera is $600,000 alone, and so it's very costly to set something up, and that's why there's now the strategy of establishing national centers. NIH has put some money into it. But there are some consideration to go to lower voltages. Richard Henderson has been advocating for this. There's, the different voltages have sort of different domains of validity and so forth. So it's working with something like 80 kilovolt or 100 kilovolt is not very explored, but you might get to the same kind of resolution. We don't know. And then the price would go down, but still, it would still be above $1 million.
Well, I think there are no other questions. I'm going to ask one, a practical question. So for the recent work you're doing on the ribosome, how long does it take? And how many EM exposures do you have to make to eventually get a structure for, let's say, 900,000 particles?
900,000.
Well, you had 870, so.
Yeah, well, but the 800,000 were used for a particular method here for the mapping of the energy landscape. But if you want to have a structure per se, then you might just need a few 10,000s, and they can be now collected very fast. And in fact, if you can, you can now essentially get a reconstruction in a few days. Including the data collection and the image processing, it can all be done. As I mentioned, ribosome, we know exactly how to do a sample preparation. We can really go without any problem.
Does it require a lot of computing power?
What?
Does it require a lot of computing power, or you have very specialized hardware for it?
These are all specialized now. These are GPUs that already hardwired for particular functions. So, so this is And it's relatively cheap now. These GPUs are relatively cheap.
Yeah, I understand Google deploys them by the hundreds of thousands, so yeah.
Hmm.
Anyway, okay, if there are no more questions, let's thank our speaker. And I'm going to present the speaker with a small token of thanks for giving this talk and coming down here. One is a copy of the announcement of the talk, signed by all the members of the General Committee on behalf of all the members. And the other one is that reprint of the bulletin of the Washington Academy of Sciences, Volume 1, covering the period from 1871 to June 1874, having the minutes of the first meeting, the president's address, a list of the members, a method for computing pi to 30 decimal places, and other really interesting historical information. So thank you so much.
Thank you.
Joachim. Before we go, a few closing announcements. Please remember that PSW depends on members and sponsors. If you are a member and you haven't paid your dues yet, please do. Whether you have or have not paid your dues and you're a member, please consider making an additional donation, sponsoring a lecture series, and or volunteering to help carry out the Society's activities. And if you're not a member, please join. You can apply for membership via the PSW website. Just go to the home page and hit the join button, and these various pages will come up, which you can fill out. I'm sure anybody who's here is interested in science can navigate these relatively uncomplicated web pages. And when you submit your application, it will be quickly considered. And if you have a genuine interest in science, you will be accepted to membership. Keep in mind that PSW is a nonprofit educational organization, still tax exempt under Section 501c3 of the Internal Revenue Code, and contributions are tax deductible to the extent permitted by law, and whoever happens to be in power. All PSW members are in good standing. Who are in good standing may wear the PSW rosette. If you wish to purchase one, you may do so at the rosette table at the back of the auditorium, or you can do it online. It's $15 plus tax. Our next lecture will be the 2421st meeting of the Society. It will take place right here in the Powell Auditorium on March 6th, 2020, and the speaker will be Rajesh P.N. Rao, who is the Yun Wang Professor in the Department of Electrical and Computer Engineering and the Director of the Neural Systems Laboratory at the University of Washington in Seattle. And he'll be speaking on when AI joins the human brain co-processors for restoring and augmenting human function. Rajesh is a world expert on directly connecting brains to computers. Not quite at the level of the Borg, but he actually does this. And it's an area undergoing rapid development with potentially profound implications for human individuality and for society as a whole. Siri is a merely verbal communication. Rao's work involves more intimate brain-computer interfacing, not only directly between one individual and a computer, but also, and he's done this, many-way connections between multiple subjects and computers. Oh, the date is wrong on the slide. Oh, my goodness. I apologize. It's not 21st February. It is 6 March. Ignore this slide. Go to the website. The lecture is sponsored by PSW member Adarsh Deepak. On the 20th of March, for lecture 2422, we have a kind of companion talk by Henrik Christensen, who is professor of engineering and director of the Contextual Robotics Institute at the University of California at San Diego. And he'll be speaking on robotics, and more information should be posted to the website this weekend. This lecture will be sponsored by PSW Science member Erica Kane, who's here in the audience. So we say thank you, Erica. Excellent. The 2,423rd meeting will be on April 3rd, 2020, and I'm waiting to hear from the invited speaker that they can do the talk. So more on that later. On April 17th, 2020, for the 2,424th meeting, we have Wendy Friedman, who is professor of astronomy and astrophysics at the University of Chicago, and she will be talking on measuring the Hubble constant and the fate of the universe. There is some discrepancy in different methods for measuring the Hubble constant, which is an important parameter of what is happening in the universe and what ultimately will be its fate. And so it should be a very interesting talk. It's a kind of companion talk to the talk that was given by Adam Rees about 15 years ago on a similar subject, how fast is the universe expanding, or how much faster is its expansion accelerating. This talk is sponsored by PSW member Bob Terry. On May 1st, we have Rob Bertrand, who is Chief Scientist for Food Safety at USAID. He'll be speaking on golden rice. How many of you know what golden rice is? So you will all be here to hear this talk. So it proves to be a very interesting subject, very interesting mixture of science, politics, fear, disease, all the things that make for a really good, interesting evening. So I hope to see you then. And then finally, we come to the Henry Dinner and Address for 2020, and that will be meeting number 2,426. The speaker will be Shep Dolman, who is the founder and director of the Event Horizon Telescope Consortium and an astronomer with the Smithsonian Astrophysical Observatory, and also senior research fellow at Harvard University. And no surprise, he'll be talking about imaging a black hole. Although you can't really see a black hole, but you can see the effects of a black hole. And they've actually done that by putting together a team of many astronomers and observatories in a rather spectacular result. So I commend you to come and hear him. Please check the PSW website for updates. Please note that PSW is run entirely by volunteers, no paid staff at all. So let's thank our crew for tonight. James Heelan for reading the minutes, Cameo Lance for managing the room, Ann McQueen for managing the live chat and rosettes, Robin Taylor hiding back there behind the audio rack for the video setup and breakdown, the live stream, and for directing the video. Jared McQueen and Connor Nixon on the cameras. Noah Block, an all-around good guy, didn't have a specific task tonight. Our red, blue, and black mic runners, and our post-lecture wranglers, Lloyd Mitchell and Robert Thompson, who should not be overlooked because there's a lot of work to break down all the equipment, pack it up, and get it in the car. Please note that the social hour ends promptly at 10:30. Please use the side entrance to exit the building. And one more thing. Please leave your glasses, plates, napkins, crumpled up paper, etc., not on the stage, not under your chair, not on the tables that say don't put anything here. There are some oval stations for such things. Please put them there. And I will now accept a motion for adjournment of the 2,420th meeting of the Society to the social hour. We have a few motions. Do we have any seconds? Well then, the meeting is adjourned to the social hour.